morning or good afternoon, and welcome to our quarterly Scleroderma Forum podcast. I'm very fortunate today to be joined by Professor Yannick Allanore, a key opinion leader in the field, that will discuss with us two papers that have been selected by the Scleroderma Forum Steering Committee for their interest and their potential impact on where we're going next. The first paper we discuss today is a real-world efficacy and safety of tofacitinib on the progression of skin sclerosis in patients with scleroderma, an analysis of a national multicenter data. Yannick, welcome to the Scleroderma Forum.
Thank you for the invitation. I'm very happy to be part of this exercise, and we have selected two nice papers for today's discussion.
Indeed. So this first one is on JAK inhibitors. Now, for some of you will be seeing this on YouTube, some will just have sound. So I will describe a bit what this paper has found and published, and then we will discuss with Yannick what are our own thoughts and consideration. First of all, the objective of this paper was to do a post-hoc analysis of the long-term effectiveness and safety of tofacitinib in patients with scleroderma. And it's not from a trial, but it's from real-world data of a national cohort in China. So, of course, it has all the pro and cons of real-world data, including the post-hoc analysis. And the inclusion criteria that they were applied to the cohort were patients fulfilling classification criteria, treated at least 3 months, with at least 2 recorded MRSS and PGA scores. Then, to put these results in context, since it's not a randomized controlled trials, the authors made a propensity score matching 3-to-1 with people not on tofacitinib, and so that they could identify patients that they looked similar as far as the other clinical features and compare what was their outcome. And then, of course, they did the longitudinal changes analysis in MRSS and PGA. Overall, 80.6% of patients were female, as we expect. They were 46 years of age. And of course, propensity score matching is a very powerful method to match populations. So there were no significant changes in the external comparator. What the authors have described is that tofacitinib, at least 3 months treatment with tofacitinib, was associated with a greater and faster reduction of MRSS compared to the propensity score matched external control, with a 2.6-fold difference in decline over time. And this, of course, it was significant. They also observed a greatest benefit when tofa was associated with standard immunosuppression, in particular methotrexate, where this fold was even higher, and it was reaching 7 points different. So altogether, from real life, they showed that tofacitinib could be useful, and of course, it did not have any issue in safety in a 3 months cohort. And so they proposed that further research should be done to evaluate its efficacy. And this was the paper. It was published only recently on JM ACAD Dermatology 2026. And here it is for your consideration. Yannick, what do you think?
It's very interesting because the sample size is very large for this network, and we need data about JAK inhibitors. It's a class of drugs that we know very well in rheumatology, but that is expanding very much in dermatology. Many inflammatory conditions are now investigated with the use of JAK inhibitors. And it's a pity that systemic sclerosis was not more carefully investigated with these drugs, because it came out that the warning regarding safety have somehow limited the classical development in systemic sclerosis, and nevertheless, the biological properties of these compounds would make sense in the disease. I think so far, Francesco, we had a small phase 2 trial, and there is an open label coming from India but
Yeah.
also suggested some trends of benefit, but it's not solid enough to think that we can offer this drug to the patient. And of course, these are not registering trials, but it's showing a direction. I think first it's reassuring regarding the safety because we cannot see any warning signals from these studies, but the content is stimulating, but it's very preliminary. For example, only 3% of the patients from the network received tofacitinib. It's a very small, probably highly selected population of patients. And I was also, let's say, a bit disturbed by the fact that many patients had the limited cutaneous subset. Even it makes sense regarding scleroderma as a whole for being treated. If MRSS is the primary outcome measures, I don't know if you agree, but it's challenging to use MRSS, which is not validated in this
Of course.
subset.
I entirely agree. Entirely agree. So very small population, limited value of MRSS in limited disease, still stimulating. I think the most stimulating um, element of this study is that tofacitinib will come out of patent very soon. And if I reflect on the history of mycophenolate that became standard of care without a proper trial, once tofacitinib will be very accessible, there is the chance that more and more data will start to build. And you're right, this warning on the potential long-term safety is something to consider, also because of the increased cardiovascular risk of our patients. But I think it's promising because the one thing I learned from scleroderma is the role of type 1 interferon and Jack is right upstream there. Jack can also influence NF-kappa B signaling. So I think the biological rationale is there. The limited study that was published by Allanore showed that there is a biological signature associated with the use of JAK inhibitors. So I think it has potential. It could be next mycophenolate.
Yeah, it could be, but yeah, from a biological rationale, it is strong. No doubt about this. Of course, the selectivity of the compounds may influence the outcomes. Tofacitinib is a pan-JAK. Is it the best drug? It was probably the first use in this population, so it is the reason why this drug was mostly investigated, but we will need data with the other JAK inhibitors. Um, I have to say that I can understand why MRSS was chosen because it's more consensual, probably more systematically collected. But I would be very curious to see the non-skin effects of
Of
these
course.
drugs because
Of
for a
course.
systemic disorder, and we know the burden of the other complication of the disease, we would really like to see what have been the outcomes on,
Yeah,
let's say, digital ulcers, but of course, lung fibrosis and some
absolutely.
other component. And also the joint, because there is no doubt that JAK inhibitors are good for the joints, and we know data
Absolutely.
from ARI. This is part of the scleroderma phenotype. And looking more specifically at the arthritis or arthralgia or the musculoskeletal component could be stimulating. Also, maybe the muscles. What do you think?
Absolutely. No, I was thinking exactly that. Now, we are here today as part of Scleroderma Forum, and we're very grateful to CISA Scleroderma Forum to give us this time. But we wear many hats, and the hats, one of the hats that I'm most close to is the work that you introduced me to in USTAR. Now, the WSF USTAR is really a gold mine and is a brilliant network. Do you think that once this drug becomes generic in all countries, the community could step up to do a prospective study, moving from clinical projects, from analysis of data to prospective collection? There is this very clever randomized within cohort design when you can propensity match and randomize patients to mycophenolate of tofacitinib, because there isn't I'm saying Euster, WSF Euster is because with this drug becoming generic, we will not find any sponsor that will do the trial. So the only way to do it is the community to take it on our shoulders and do it. What do you think? If there is any
But
young investigator listening, come and talk to Yannick.
yeah. The issue is that if we remain on the routine use of the drug, it still will be given to very selected and refractory patients, and probably it's not giving the best opportunity for the drug to show its efficacy. So what we would need would be a trial, a real trial, without the cost of the drug, but still the cost of the infrastructure, randomization, standardized monitoring, and so on, all what we know perfectly. But this is what we would need to be definitely confident, because still there are so many limitations. But if with these kind of preliminary approaches we are seeing stimulating data, maybe it could be inspiring for building a real trial.
Yeah, absolutely. Thank you. Thank you so much. Stimulating. Now, our next paper is close to this topic because it explores, in my opinion, a potential source of JAK1 activation. It is neutrophil reprogramming underlying vasculopathy and lung disease in systemic sclerosis.
It's a nice selection. It's a study coming mostly from Italy. Systemic sclerosis is definitely complex. You know the triad of endothelial cells, immune cells, fibroblasts, but it's probably more complex than that. And neutrophils have not been much investigated so far in that context, even if from a biological point of view, it could have a role in different aspects of the disease. Of course, oxidative stress, the specific netosis, which is a trap for the extracellular aspect, and that can have a role, for example, in interferon biology, which is interesting in the context. And also neutrophils can produce proteases and some other molecules that could have a role. So there is a rationale for neutrophils to have a role in systemic sclerosis. Data so far are less compelling than in lupus, and the investigators were very clever to enroll 3 groups of patients, as you can see. 39 patients with systemic sclerosis, 39 healthy controls, but also patients with lupus. And they did some classical initial biology looking at circulating cells, neutrophils, their activation, their profile, their phenotype. Then they move to collecting extra cellular vesicles to use them to see their effects on the communication with the neutrophils. And because they identified some stimulating data, then they were more ambitious, and they used these vesicles to inject them to different mice models, like immunodeficient mice. And then they also used transgenic mice that were do not express RAGE receptor. And altogether, the summary is that they are showing differences between lupus and healthy controls regarding activation, autophagy, and Type2 expression, and netosis also, with more activation in the context of systemic sclerosis. And here on the right, you can see the results in the animal models, where you can see the effects regarding Type2 expression of the neutrophils after injection of extracellular vesicles coming from patients with systemic sclerosis. And then, because they identify a specific role of each MGB1 molecules, they block the receptor by looking at mice invalidated for this receptor, RAGE, and you can see that the effect of the vesicles is disappearing. showing that the axis of each MGB1 RAGE and neutrophils can have a role in the context that we are interested in, which is activation of neutrophils and interaction with the other aspects of systemic sclerosis. So I think it's fine. So the first comment is that what is missing is a scleroderma models, because here it's only biology, cell phenotype, cell production, but we are somehow far away from systemic sclerosis. But it's a clue to investigate this pathway in scleroderma models. So how do you see neutrophils in that context, Francesco?
Yeah, I think it's stimulating sort of finding. Neutrophils are the army of the immune system. They come there. They are very differentiated cells. They are terminally differentiated cells, and it's unlikely that they have no involvement in a disease where we know there is an endothelial cell activation. The angle of looking at their activation in the context of EV is original. They're very sensitive to extracellular vesicles. The model, you're right. I think uh, in in the lack of a better models, the authors did is all they could do. To me, this shows that extracellular vesicles from patients can contribute to activate neutrophils. How that contributes to scleroderma is a different story. But there is, with all the talk about type 1 interferon and JAK inhibition, NETosis could be a very good source of that type 1 interferon response. And we know that the endothelial cells in scleroderma are activated. So it is plausible. I wouldn't say that this gives any proof, but it suggests a plausible hypothesis that would need to be tested.
What is, I think, challenging with autophagy is that there are data supporting that this can be protective or it can be harmful according to the context, the stage, the levels of activation. And this is, of course, making this axis and pathway more challenging in the context of systemic sclerosis. But in this disease, we know that disease duration is particularly critical. And it could be interesting to investigate the effects according to the stages of the disease, because
Absolutely.
maybe the effect might be different at the early, when there is the immune storm at the very beginning of the early diffuse subset, and it might be different when we are more at the stage of the remodeling and for example, proliferative vasculopathy. So
Yeah.
this would deserve to be investigated, yeah, but we know that it's not easy, because it means
No.
selection of the patient and selection of the models, and of course
Yeah,
more
but
experiments.
you're right. But now that I was hearing you commenting, I think, and if there is people listening that want an idea, there is a large data set out there from skin and from lung of single cell RNA-seq, of bulk RNA-seq. And what this paper shows to me is that RAGE and TIE2 could be associated with NETosis. So if I think the first step that we could be done in silico is just to look at the expression of RAGE and correlate it with type 1 interferon to see whether there is any link between, or TIE2 for this matter. So I think this, I can see why the steering committee selected this paper. Is stimulating to say there might be something we're not considering in our
And
entire
yeah,
set.
the last point, because you raised the interesting role of EVs in that context. I think it's another piece of work supporting that platelets can have a role in this disease, with some interactions with some other domains, because EVs are mostly coming from the platelets, and it is another piece of work supporting that release from platelets can have a role in this complex disease.
No, you're right. You're right. So there is so much out there. The more every time you have half an answer, you really have three questions. And for the people listening, we really would like you to engage. If you have ideas, questions, if you would like to do something and you don't have the tools, reach out. There is so much that we could do, and we need more brain power. With this said, and talking about brain power, I would like to thank our brain today, Professor Allanore. Thank you for your comments, stimulating us as usually. And I would like to thank you all for your attention and stay tuned. We'll come back with a podcast next quarter. Bye, guys.
Thank you. Have a nice day. Bye bye.